Hey guys, and welcome to this video of organization. If you're new here, make sure to click that subscribe button. Okay, so we'll be learning about enzymes and their digestive system. So to start off with, we need to talk about food groups that are digested in the digestive system. So the first one is carbohydrates. So carbohydrate is used in the body is an energy source used in the body for respiration. And carbohydrate is a long chain of sugar. So starch is found in many food types such as bread and pasta, and it's a type of carbohydrates. So for food tests, iodine is a solution that can be used to test for starch. So iodine is a yellowish brownish solution, and if starch is present, then the food will turn into a blue or black color. To test for sugar, Benedict solution is used. It starts with blue and we need to mix it with the sugar sample and heat it. If there's low level of sugar, you'll get a green color, and if there's a high level of sugar, you'll get a brick red color. Another food group broken down in the digestive system is protein. Protein is obviously used for growth and repair, as we have learnt in the last video, and proteins are also chains of amino acids. And a burette test can be used to test the presence of protein. So for the burette test, the chemical is blue. So if there's protein present in the solution, it will go either pink or purple. So the last group that is digested in the digestive system is the fat and oil group, which is scientifically known as the lipids. We need lipids because cell membranes and some of our hormones are made from lipids. So they get broken down or digested into 1 glycerol and 3 fatty acids. To test for the presence of lipids, we can use 2 chemicals, Sudan-3 and ethanol. So ethanol is the easier way to test if lipid is present. So ethanol is usually put into the solution of the food and it's mixed around. So if lipids are present, then the solution will go cloudy. For Sudan-3, it's a red chemical which is mixed with the food sample, and if lipids are present, then there will be separation in the food. So the top layer will go bright red, and then the bottom will say whatever color the food was. So if it goes bright red, that means lipids are present. Okay, so digestion. What is the meaning? So digestion is the breakdown of large insoluble molecules into small soluble molecules that can be absorbed into the blood. So this breakdown is sped up by enzymes, as we learnt in the last video. So let's look at how the digestive system works to break down these 3 groups. So the first bit is the mouth. So over here is where mechanical digestion and chemical digestion occurs. So mechanical digestion is when food is physically broken down by teeth. But over here we're more concerned about the chemical digestion. So chemical is when the food is broken down by enzymes. So the enzymes involved in the food, the enzymes involved in breaking down the food in the mouth is the it's called the carbohydrase enzyme. So the enzyme involved in digestion is in the mouth, and it breaks down carbohydrates into sugars using an enzyme called carbohydrase, and this particular carbohydrase enzyme is called the amylase enzyme, so you can see over here. So from the mouth, the food moves down into the esophagus and down into the organ which is called the stomach. So here the proteins are broken down, and the proteins get broken down into amino acids. So over here you can see. And the enzymes that break down protein are called the protease enzyme, and the particular enzyme that works in the stomach is called pepsin. So the stomach is lined with glandular tissue that releases the enzymes, and it also releases hydrochloric acid. So the hydrochloric acid isn't used to break the food down, instead it's used to kill the germs in contaminated food and drinks, and it also creates the optimum condition for enzymes to work, which is pH 2. So the food has now been partially digested and it moves to the small intestine. So here the small components of food is absorbed into the bloodstream. So here the lipids get broken down as well as the other two food groups. So lipids are broken down by the enzyme called lipase. You can see that over here. So lipase enzyme breaks down lipids into one glycerol molecule and 3 fatty acids. So bile is also a chemical which helps with digestion, and it's made in the liver. So after it's made in the liver, it's stored in the gallbladder, which is this little greenish type of organ. And when bile is needed, it is sent into the small intestine through a tube called the bile duct. So lastly but not least, this yellow leafy type of organ is called the pancreas. So the pancreas is basically an organ which makes all three groups of the enzyme, so the amylase, protease and lipase enzyme. So now let me just briefly explain how bile is involved in lipid. And flowing through the small intestine, you can see a clump of lipids up here. So you can also see here that one individual lipid is made from one glycerol and 3 fatty acids. So the bile that is made in the liver and stored in the gallbladder is then sent into the small intestine via the bile duct, and then it helps to break up the larger clumps of fats or lipids into smaller clumps of lipids, and we call that emulsification. So over here you can see the bits of green dots, which is the bile, and we can see that the bile has emulsified the fats into smaller clumps. So basically emulsification increases the surface area of the lipid, so it gives more access to the lipase enzyme to work, and it's easier for the enzyme to work. So once the lipase has acted on the group of lipids, you now have small soluble pieces that can easily be absorbed into the blood. We've already gone through the structure of villi in the last video, so if you need to go through that again, then go check my video out. I'll keep the link in the description box. Okay, so now this is just a summary of everything we've learned, so it's easier for you guys to understand. So the enzyme carbohydrase or amylase breaks down carbohydrates into sugar, and it's produced in the small intestine, pancreas and salivary glands. The enzyme protease, which breaks down protein into amino acid, is produced again in the small intestine, pancreas, but then in the stomach. And the enzyme lipase, which breaks down lipids, which is basically fat, into fatty acids and glycerol, and is produced in the small intestine and pancreas. So we can see here that all three are produced in the small intestine and the pancreas. Okay, so on to the exam questions. Make sure to pause the video and have a go. Okay, so this is the answer. So the lines which have the 1 mark next to them is basically the ones that will get you the marks. Don't worry if you've got this wrong, just make sure that you've written those important keywords such as emulsifying and then the name of the enzymes. Okay, that's it. Thanks for watching, guys. If you liked the video, make sure to